In the last video, we explored some fundamentals of genome sequencing, the process by which we generate the genome of a particular organism. Now in this video, we're going to look at what can we do with that genome sequence information? What are some of the applications of genomics? So let's go ahead and take a look at that. So once we generate a genome sequence, generally one key piece of information that is going to be of use from that genome sequence is going to be the specific genes that are encoded by that gene sequence, because ultimately the genes are going to be transcribed and translated into the proteins, and the proteins are going to determine a lot about the function and health of the cell. So what we do is we take the genome sequence, and this could be either the complete entirety of the genome, it could be a draft genome that contains contigs that represent portions of the genome, or it could be just a very small targeted portion of the genome that we are interested in. So either the global genome or a targeted genome sequence is taken, and from there, a variety of tools exist depending upon whether you are dealing with a prokaryotic genome, a eukaryotic genome, and specific what type and species of organism you're dealing with, to predict the individual open reading frames, or the ORFs for open reading frames. So generally the open reading frames are predicted, and this is done computationally using so-called bioinformatics tools. And with the prediction of the open reading frames, the other way that we could think of that is that we're predicting what genes are encoded by that genome. And by predicting what genes are encoded by a genome, we looked earlier in the semester at how we could take a codon table and use the sequence of a gene to propose the sequence of the protein that would correspond to that. And that can be done both manually and in an automated fashion. So what I'm going to list here is, in virtual terms, transcribe and translate the gene sequences into the predicted protein sequences. And even in an organism as simple or seemingly simple as bacteria, some bacteria have upwards of 5,000 different genes that are predicted by analysis of their complete genome, and hence 5,000 or more different proteins from that relatively simple bacterial genome. When we look at genomes that are larger and more complex, the number of genes goes up dramatically. Although do keep in mind that with eukaryotic cells, there are some introns mixed in with the exons, and those introns are not transcribed and translated into proteins. So that creates some extra bulk in eukaryotic genomes that aren't present in bacterial genomes. But in bacterial genomes, there are no introns versus exons. Instead, what you see is what you get. The open reading frames correspond directly to the genes that are transcribed and translated into the proteins that the organism is able to produce. Keep in mind as well that just because an organism has a particular gene that encodes for a protein, that doesn't mean that that gene is constitutively expressed. That doesn't mean, in other words, that that gene is constantly being transcribed and translated into a protein product. Instead, the gene can be repressed or activated at different stages of its life cycle. And so just as our proteomics analyses had some limitations in terms of what information they can give us about the state of a cell, genomics has some limitations as well, because genomics is going to tell us what genes are present. It's not going to tell us whether those genes are being expressed into proteins or not at a particular time point. So once we have that particular set of proteins that the organism is capable of producing from those predicted genes, what are some of the applications of this genomics information? We are going to look at a couple of those possible applications, one of which is that genomics has revolutionized our understanding of bacterial species. Prior to genomics, we relied upon such techniques as gram staining to distinguish between gram-positive and gram-negative bacteria and establish some morphological traits such as the shapes of cells and things of that sort. We also had techniques for sequencing very limited portions of the genome, such as techniques for sequencing 1,500 nucleotides of the genome, and using that as a way to describe the relatedness between different bacteria. But as it turns out, those limited DNA sequencing techniques or morphology-based techniques left a lot of information unclear about how closely related or distantly related different bacteria were. And so as a result, the genome sequencing of bacteria, which is generally straightforward relative to sequencing human genomes and other more complex genomes because bacterial genomes tend to be smaller, we can understand better the processes of bacterial speciation and how related different strains of bacteria are. And this can be useful both in an environmental context if we are looking at different habitats and trying to compare and contrast the bacteria that are present in those different environments. We can complete the genome sequencing of those bacteria and compare the genomes of organisms in those different environments to understand how related the bacterial species are that inhabit one environment versus another. Also, in terms of clinical relevance, we can use genome sequencing as a tool to evaluate different bacterial pathogens that are from the same species, where some pathogens may be more virulent than others or more capable of causing serious illness than others. So we can look in clinical settings at comparing bacteria from the same species for different impacts on their host cell. Where if we understand the genome sequence of a particular bacterial pathogen, we can recognize and predict whether that is going to be a particularly deadly strain of bacteria or whether it's going to be a relatively harmless strain of bacteria. Another application of genomics that has come into much more prevalence in the last few years and will likely continue to become increasingly relevant as we move forward in time here is the analysis of human genetic variants. So what do we mean when we say a human genetic variant? What we're looking at when we evaluate a human genetic variant is we're looking at variations in individual genes among different individuals. And what these comparisons of a particular gene amongst different individuals has enabled is it's enabled us to pinpoint the genetic origin of a variety of different diseases that previously weren't known to have a genetic origin, such as some breast cancers have now been linked to specific genetic variants. And we would not have had that understanding prior to this. And so now there's a variety of companies such as 23andMe and others that will sequence snippets of a person's genome in order to look for variants that may correspond to different disease states or may correspond to different allergies and a variety of other things. And as time goes on and we generate increasing numbers of human genome sequences for people with a variety of health conditions, we generate dramatically improved capabilities to use these human genetic variants as tools for detecting disease or detecting things such as athletic performance and a variety of other traits that differ across different humans. And so by gaining a better understanding of these human genetic variants, meaning small variations in genes amongst individuals, where one person might have a gene that had an arginine residue at a particular, that encoded for an arginine residue at a particular position of that gene, whereas someone else encoded for an alanine at that position, and we could link that to different traits, we would be able to create predictive ability where in the future we can look at a human genome and predict a variety of things about that particular individual. So this analysis of human genetic variants has been really useful for predicting the likelihood of a person developing a particular disease. It has also been really useful in terms of determining the best course of treatment for diseases, because we can generate a body of information where we can link the outcomes of treatment with a particular drug to particular genetic variants that individuals had to really customize and personalize the field of medicine. And this is an area of genomics that is likely to continue to dramatically grow and increase, particularly as the cost of genome sequencing continues to decline, as we saw in the video prior to this. So there's a huge variety of applications of genomics that we can link to our previous understanding of proteomics and metabolomics to gain a very complete, elaborate set of information about whatever model system we are studying. And so increasingly commonly, the fields of genomics, proteomics, and metabolomics are not used as individual isolated tools and techniques, but instead are merged together to create a more comprehensive understanding of a variety of biological traits. And we can link those together since we consider that genes are transcribed and translated into proteins, and those proteins are the enzymes that catalyze the chemical reactions leading to the small molecules detected by metabolomics. So although up until now we've sort of presented genomics, metabolomics, and proteomics as three separate things, in actuality they are becoming increasingly integrated, where a person would take a treatment and control sample, isolate the DNA from that sample to use genomics methods, isolate the proteins from that sample to use proteomics methods, and isolate the metabolites from that sample to conduct metabolomics analyses, and combine those three together to gain a much more comprehensive understanding of the status of the treatment relative to the control than would be possible using any one omics technique alone. And so that's why we've covered those three together, is they do really work in synergy with one another, where each of those is focused on a different layer of understanding related to a different type of biomolecule. There are also extended types of omics, such as lipid omics, looking at the lipids present within a particular sample type, or glycomics, looking at different carbohydrates present within different types of cells. But our understanding of genomics, proteomics, and metabolomics that we've developed in the last several sessions gives us an overview and understanding of the potential that these various chemical techniques for understanding different types of biologically relevant molecules provides in diagnosing and treating disease, in understanding organisms that live in different environments, and a plethora of other applications that are likely to continue to grow as time goes on.